Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're going to continue our lesson today talking about meiosis. Specifically, we're going to talk about the stages of meiosis 2. So let's go ahead and get into it and see what we're talking about today. So just a quick overview of what is meiosis. Well, meiosis is a specialized type of cell division. We're making gametes here, okay? Gametes are egg and sperm cells, and this is going to happen in the reproductive organs, okay? Meiosis is often referred to as a reduction division because it reduces the number of chromosomes by half. We want our egg and sperm cells to have half the number of chromosomes as usual so that when they come together, now we have the full 46 that we're supposed to. Okay, when they come together, that's known as fertilization. Okay, during fertilization, two gametes combine to form a zygote, which is diploid. The first thing humans ever are when you're first made in your mother's womb, you're a zygote. A zygote is a one-cell large human or organism that has all the chromosomes that's supposed to. So egg and sperm come together, now you're a zygote. Okay, you have all the chromosomes that you're supposed to. Okay. This is a form of, we use this during sexual reproduction, okay? It involves a cycle where meiosis and fertilization alternate to maintain chromosomes number across generations, okay? So we use this for sexual reproduction. It's how we make gametes so that we can reproduce sexually, okay? Today we're really talking about meiosis 2, okay? And so just as a quick overview and reminder, meiosis is actually divided into two phases. Okay, we have meiosis 1, which we talked about last time, and we have meiosis 2, which we'll try to talk about today. Okay, the outcome for meiosis is that we want to make 4 haploid cells from 1 diploid cell. Okay, and that's in both men and women. Okay, mitosis results in 2 genetically identical diploid cells from 1 diploid cell. So there's a difference here, right? Meiosis, we make 4 cells that are different from each other, and in mitosis, we make 2 cells that are identical. Okay, so there's kind of a difference there. Okay, so we talked about meiosis 1 last time. We'll pick up with meiosis 1 today. Okay, after meiosis 1, this is where we're at. So the resulting cells have half the number of chromosomes as the original cell, but each chromosome is still present in matching pairs. So we're really talking about the bottom part of this picture today. Okay, what's happened up to this point, just as a reminder, is that those homologous chromosomes line up. When they line up like this is called a tetrad. The tetrad is going to cross over, so we're swapping some of our genetic material, okay? Then in metaphase, they line up in the middle. In anaphase, they get pulled apart. And then in telophase, we make 2 cells. Okay, so that's meiosis 1. And then we're starting down here with meiosis 2 today. So let's see what's going on. So just a overall overview of meiosis 2. Here's what's happening. In prophase 2, you'll notice that for meiosis 1, meiosis 2, and mitosis, there's a lot of similarities in what's happening in these steps. So in prophase 2, chromosomes are going to condense again, okay? And spindle fibers are going to form and the nuclear envelope breaks down. That's happened in prophase 1 and in mitosis as well, okay? And metaphase 2, just like in the other times that we've seen this word, the chromosomes line up in the middle. So the main word to remember for metaphase is middle. They line up in the middle. Okay, then in anaphase 2, now notice that this says sister chromatids are pulled apart to opposite sides of the pole. And in meiosis 1, it was the homologous chromosomes that are pulled apart. But in meiosis 2, it's sister chromatids that are pulled apart. So we're kind of splitting that X shape. We're splitting that chromosome and pulling each side of the chromosome to 2 new cells. Okay, and then in telophase, this is going to also include cytokinesis 2. The nuclear envelope is going to reform around each set of chromosomes, and the cell is going to divide, resulting in 4 distinct haploid daughter cells. Okay, important to note here that DNA replication does not occur between meiosis 1 and meiosis 2. Okay. And an overall summary, at the end of meiosis, 4 daughter cells are produced, each with half the number of chromosomes that they're supposed to have, that the parent had. These cells are genetically unique, okay? And when they fertilize, that's when the egg and the sperm cell come together. Now we're going to make a diploid cell, okay? So what's happening here. So in meiosis 2, again, remember that there is no interphase between meiosis 1 and meiosis 2. Okay, and also a reminder here that meiosis 2 is like mitosis in that it separates sister chromatids, but it results in 4 haploid cells rather than 2 diploid cells. So there's some similarities and some differences here. Okay, here's a breakdown of something we talked about last time known as non-disjunction. Okay, non-disjunction can happen in anaphase 1 or in anaphase 2, and that's what this is supposed to be showing us here. So let's kind of take a closer look at what's happening, and we'll talk about it in a little bit more detail. Okay, so over here on the left, this is what can happen. This is non-disjunction. This is what it would look like if it happens in anaphase 1. So notice up here we've got 2 pink chromosomes and 2 blue chromosomes. Okay, they are supposed to be separated and go into these different cells. But we've had something known as non-disjunction happen. Non-disjunction is when the chromosomes don't separate the way that they're supposed to. So now when that happens, this cell does not get any of those pink chromosomes. So it's missing chromosomes. That's going to be really, really bad. This gamete's basically not going to work the way that it's supposed to. Over here, this one has too many chromosomes. And this is still bad, but just not as bad as missing chromosomes. And as the result of this, you can see that these 2 cells, because that pink chromosome came here and then they separated again, this has 1 extra chromosome over here, and these have 1 less chromosome over here. So this is non-disjunction in anaphase 1. Over here, this is non-disjunction in anaphase 2. So now what's happening is it's actually separated correctly up here. We have 2 pink and 2 blue. They separate, and you can see that we have pink chromosomes over here and blue chromosomes. So they separated correctly in anaphase 1, but then non-disjunction happens in anaphase 2, and you can see that the result is very similar here. Now we're ending with 1 chromosome that has an we're ending with 1 cell that has an extra chromosome, 1 cell that has a missing chromosome, and 2 normal ones. So non-disjunction is worse if it happens in anaphase 1, but it's still pretty bad if it happens in anaphase 2. Okay, if we're not having the correct number of chromosomes, this is going to lead to some problems down the road. If you're missing chromosomes like these, if this egg or sperm cell is part of that fertilization process, that organism is not going to survive because it's missing information that it needs. Okay, if one of these egg or sperm is used in that fertilization, the offspring may survive, but it'll have some serious genetic defects, okay? So after that non-disjunction, remember that happens in anaphase. So now we're in telophase 2, okay? This is the last part of meiosis, okay? In telophase 2, what's happening here, just like in mitosis, a new nuclear envelope forms around each set of chromosomes, and then cytokinesis is going to happen. Remember, cytokinesis is division of the cytoplasm, so the cell undergoes a second division. This is going to result in 4 haploid daughter cells, and each of these daughter cells has chromosomes in a single condition. They're not doubled. These cells are gametes. They're either sperm or egg cells. They can be ovules or pollen grains. Every organism does this if they're reproducing sexually, so this could be in a human, this could be in an animal, this could be in a plant, but it all results in the same thing, those 4 cells that are all haploid from each other. So here's just kind of a breakdown of what's happening again. Up at the top, this is meiosis 1, right? And what's happening here, some key things that I want to point out is you really want to be able to tell the difference in between homologous chromosomes and sister chromatids, because that's really going to help you determine what phase you're in. Okay, right now they're labeled, but you can imagine how tricky this would be if you didn't have the words right next to it. Okay, so what I really want us to notice, look here, you see how we have these 2 chromosomes that are side by side, 2 X's that are right next to each other? These are the homologous chromosomes. We only have homologous chromosomes in meiosis 1. So if you ever see this, you know that we're in meiosis 1. Here it's prophase. When they line up in the middle, it's metaphase. And then when they're getting pulled apart, it's anaphase. Okay, it's a little different in meiosis 2 because instead of the homologous chromosomes being put in the middle and divided. We're dealing with something else, and these are known as sister chromatids. Notice that there's only one X in these cells, right? Not two, because we've divided, after meiosis 1, you divide the number of chromosomes in half. So we have less chromosomes down here. We had 2 up here, we have 1 down here. So now, during meiosis 2, we're dealing with sister chromatids, right? The sister chromatids get pulled apart. Up here it was homologous chromosomes. So that's kind of the difference in between those. So y'all did a great job. This stuff is tricky. I have some review stuff we're gonna do. So teacher, why don't you go ahead, take that worksheet and pass it out to everybody. It's just 10 questions. Why don't y'all take 5 or 10 minutes to work on it, and then when you're done, if you want a little bit of help, come back to me, unpause the video, and I'll kind of review it with y'all. So take a second to do that, and then when you're ready, come back to me and I'll do it on the board up here. All right, I know that didn't give y'all any trouble, but just in case it did, I'm gonna review those with y'all. So let's kind of take a better look at this and see if we can figure out what's going on. So we'll go ahead and get started. Y'all feel free to change or add anything that you don't have. So let's see what's going on here. All right, so number 1 says, the chromosomes that pair up during meiosis are called blank chromosomes. So what are those chromosomes called? They're very similar in size and shape and have roughly the same genetic material in them. Those are called homologous chromosomes, okay? Notice that this prefix homo, you've seen that before, homo. Homo means the same, and so these chromosomes are very similar to each other. That's why they're called homologous chromosomes. All right, number 2 says, how many daughter cells are produced from meiosis? So in meiosis, we start with 1 cell. How many do we end with? We end with 4 cells, right? And they're all going to be different from each other. All right, let's look at number 3 next. Okay, 3 says, name the growth phase of the cell cycle for both mitosis and meiosis. So what do we call that phase? The cell's not dividing, but it's growing and getting ready to divide. You might remember that that's called interphase. That happens with both mitosis and meiosis, but it's very important to note that interphase does not occur in between meiosis 1 and meiosis 2. It happens before, but not in between. Okay, great. So let's scroll down and keep it going. All right, so here's number 4. This says, this image shows chromosomes during prophase 1. Which of the following correctly describes the process shown? So what is this called? These homologous chromosomes are lining up, they're swapping genetic material, and at the end they're different than what we're started with. What's that process called? You might remember that that's called crossing over, okay? And it's called crossing over because they're literally moving from one chromosome to the other. They're crossing over to the other chromosome, okay? This is very important. It's going to increase our genetic variation and make us all different from each other, okay? All right, let's scroll down and keep this going. Okay, so number 5, this says a human zygote should have how many chromosomes inside? Okay, well remember a zygote, this is the first time an organism is made in the mother's womb. An egg and a sperm come together, okay? And for humans, how many chromosomes should a zygote have? It's 46, right? Humans have 46 chromosomes. Sometimes people say 23 pair, but it's the same thing, 46 total, right? Okay, the next one here, number 6. It says, at the end of mitosis, you should have blank cells. Okay, so at the end of mitosis, note that this says mitosis and not meiosis. At the end of mitosis, you have cells that are, does mitosis make cells that are similar to each other, identical, or different from each other? Well, they are identical. At the end of mitosis, you have identical cells. We want those cells to be exactly the same in mitosis, okay? But let's compare that to number 7. Okay, 7 says products of meiosis are genetically what? So in mitosis, they're identical, but in meiosis, they are different from one another. Okay, they're different, and we want that. All right, 8 says human eggs and sperm are blank and each has blank chromosomes. So what word do we use to describe gametes and sex cells? Are gametes and sex cells diploid or are they haploid? Well, they're haploid. So I know that these can't be right, okay? And if they're haploid, are they going to have the full set of chromosomes or half the number of chromosomes that they're supposed to? Well, to me, I remember it haploid and half kind of sound alike. So they're supposed to have half the number of chromosomes. Humans have 46, and so that means egg and sperm are only 23 each, okay? But when they come together, that's how we get a zygote with 46 chromosomes. All right, we just got a couple left. Y'all hang in there with me. Here's 9 and 10. 9 says, how are your chromosomes inherited? Okay, so where are your chromosomes coming from? Okay, any of these look right to y'all? So this one should be half of your chromosomes come from your mother and half come from your father. Okay, this is why you kind of look like either one of them. You might have your dad's eyes, but your mom's hair. Half of your genetic material comes from mom, half comes from dad. This is going to be a lot more important in our next unit when we're doing genetics and Mendel and Punnett squares and stuff like that. Great. All right, here's the last one for today. What kind of cells are made in meiosis? Okay, what kind of cells are made in meiosis? Can we make skin or liver cells? No. We make haploid gametes. And remember, gametes is just another word for egg and sperm cells. So y'all, I wanted to make sure I reviewed that with y'all because it can be kind of tricky. Your teacher has some other problems for you to work on, so make sure to give that your best effort, and I hope everybody has a great day.